So I've always had a real passion for soils, and I think we're at a critical stage in soil science. So today, I want to talk about what I term soil security. I will go on and then discuss soil biology and something I call the root of the problem. And then we'll end up in an area which I think will be one of the fastest growing areas, not just for research, not just for development, but for advisors as well. And it's an area that I call designer soils. The first thing I would like to ask, and I ask this and I show this slide to some politicians as well, are we looking after the right things? If the last blue whale choked to death in the last panda, it would be a disaster, but not the end of the world. But if we lost vital components within the soil, ammonia oxidizers, that would be another matter. When they die, we die. The problem we have as soil scientists, farmers and researchers and advisors is that we are dealing with what is probably the most important material on Earth. And it's where the food begins, as you can see on the right-hand side. But this is an issue, this is an area that journalists call MIGO. Does anyone know what MIGO stands for? My eyes glaze over. This is a serious thing because it's not sexy, it doesn't translate well onto TV or indeed radio, and apart from the odd dust storm, no one really cares. And I think in Australia we are at a precipice in terms of our ability to actually attract the youngest minds into agriculture and soil science. And I think it's incumbent on organisations such as GRDC to help the universities, the colleges, to actually have a strategy to encourage those young people to come in. And I think we're in deep trouble just now. I believe this should be the number one priority of the nation, and Jeff, who you'll hear after me and myself, are part of a new Soil Security Alliance, which takes in about 3 or 4 universities, as well as CIRO, which is asking that very important question: how much fertile soil do we have in Australia? How can we secure that resource for the future generations? Now that's still MIGO, that's still my eyes glaze over to journalists. But remember, without fertile soil we all die. And this is the nub of the matter. 99.7% of human food comes from soil. 11% of the world's land is used to produce food. In the last 40 years, 30% of the planet's fertile soil has been lost. And when it's been lost, you're not going to truck it back in. It's lost forever. And we know in history, ancient and recent, that once you lose your soil's resource, you lose your civilization. Now, I've given this talk, Soils and Civilization, for the past 5 or so years, and people just sit there going, but we're fine. We're absolutely fine. Look at us, we grow crops. Look at us, the soils are deep. Well, I'm sorry, ladies and gentlemen, it's just not true. There's many civilizations in Roman times and beyond that have thought the same thing. Has anyone ever heard of the second greatest city after Rome in Roman times? Does anyone know that city? It was called Antioch. Antioch was one of the fastest growing cities in the world, second only to Rome, and they found it about 50 years ago under 23 foot of silt. Because they didn't take care of the soil resource, the whole population died, and the civilization died before that. It roughly takes an inch of soil takes 500 years to build. Now don't worry, this is the bad news, the good news comes quite soon. I did a calculation, I asked myself, how much soil, fertile soil, have we got left in Australia? That's a really tricky thing to answer. This simple model takes in lots of assumptions, don't get too worried, but I don't think it's too far off the track. Roughly we're losing 1% of our fertile topsoil under conventional tillage. No-till is a bit different, but roughly 1%. If you start with a soil, here's the year, and here's the depth of topsoil in millimetres. If your depth of topsoil is roughly 150 millimetres and you've got only 0.5% of erosion, that's the degradation in the topsoil. At 2250, you've only got about 50 millimetres left. Long before that, we'll be long gone, because that is not sustainable. Imagine if you just took the average in the world, which is 1%. Over half will be lost within 100 years. And if you go to 5%, we're all dead very quickly. We do not put enough emphasis on this. If you look at any other industry, they look at their core values and they look at what they really need. The gold industry knows exactly how much gold they've got. The oil industry spends a huge sum of money figuring out how much oil they've got because it's core to their business. We have probably spent pretty much nothing in figuring out what's core to our business, soil. And I would commend this to you and say we need to take quick and efficient action to figure out how much we've got left. We're mining a very limited, fragile resource, and in some cases we're in deep trouble already. So this is what I call compound debt in soil. You don't have to lose much in terms of percent of your topsoil each year to actually be in great trouble within a generation. Now actually this little graph here, this line here, is probably very bad no-till. Very good no-till probably goes way up here, where actually the farmers who have introduced minimum till are doing a very good job. But the problem is, not everyone is doing that. And also, this very simple model does not take into account the loss of cheap fertilisers, salinity, decarbonisation, Jeff will talk about that, water. When we include these issues, the message just gets worse, much worse. The key thing about where we are in our society today is we know far more about it than anyone else ever did. And it'd be crazy to make the mistakes that people made in the past. And if we don't secure the limited fertile soil we have, we will not be here within several generations. This chap called Oliver de Schutter, he's got a special role in the UN now, and he has a role related to the right to food. And his summary of all of this, and this work has been presented to him, is that we need to focus our efforts on an agriculture which does not degrade the soil. I actually don't agree with that because I don't think it puts agriculture in a great light, and the vast majority of farmers have done an amazing job for Australia. Far better than many other farmers in many other parts of the world in terms of the quality of the environment they leave behind when they farm, in terms of the soil that they have to deal with. But what I would suggest is that we do need national soil security legislation to protect and improve this fragile resource. Because basically it doesn't matter what platform of delivery you've got, what new breed of plants you've got, new crazy system to kill all the pathogens in soil. It does not matter how much you invest in this, because if you do not invest in the core fundamentals of fertile soil, we won't need anything. And that is a great issue. If you want to know more about this, we've started to develop a website called soilcolumn.com, and it's one that's just went live last week. And we're busy, a few of the professors of soil science around the world are trying to make that into an easy access portal for many issues related to soils gathered throughout the world from various sources, government and journalism as well. And that's just live last week. So saying that, the good news stories are the new technologies that are with us just now. Technologies that we could literally pick up, develop within the soil science context, take out and give to the farmers and the plant breeders. I've always been fascinated by soil structure from a physical and chemical point of view. Here we have a cube of a soil, put in a CAT scan, fire higher energy x-rays at it, and suddenly we can see inside. And this is you taking a real walk through soil. You can look at the roots, you can look at the cracks, you can mathematically take them out, and then you can start to predict water uptake, water flow, water retention. You can actually predict microbial diversity from this as well. Now, Jeff will be busy talking about soil carbon. And my view on soil carbon is very simple, and I'm not going to go into much detail. Basically, without it we die. It's the fuel for microbes and processes. Crucially, it retains water, imparts structural genesis and resilience against anything that we would like to do to the soil, and it improves the mechanics of the soil system. But a real growing thing that I want you to note throughout this talk is that we cannot look at things in isolation. Even advisors cannot look at things in isolation. If you look at soil carbon without what we call the soil architecture or the structure, it's almost a waste of time. You might find things out in the lab, you will never be able to take them out into the field. If you look at these fantastic measurements you can do in soil architecture in the lab, without understanding soil carbon and soil water, you'll go nowhere. And the great thing about the soil structure is that really it controls all the rate processes in the soil, be it water flow, gas flow, or microbes. And with soil moisture, it really controls everything in the soil. And what we have is soil carbon acting as the fuel, and the architecture is the ignition and gearing. And the key thing I would like to say is that those interactions are the things that we must look at. If there is a notion out there that you can have a centre in soil physics, a centre in soil chemistry, and a centre in soil biology, the only thing I'll say to you, that will fail, and that will fail quickly. It doesn't work that way in the soil, and the farmer doesn't look at it that way. You have to have centres where they can combine the expertise and take that on, both in the R, D, and E. And that's the one thing that we're trying to do at UNE. Not just go for soil biology, soil physics, and soil chemistry as separate, but getting a multidisciplinary team up. When I was doing my PhD in Aberdeen University, every paper that came out of Australian soils, we would hurry to look at. The best in the world, absolutely the best. And they've provided a fantastic foundation of what the farmers are doing now out in the field and our basic understanding of soil. When I look at the papers coming out now, I immediately go to France and the UK. We don't even look much at the Australian side. The degradation of the capability and the skill set in Australia soil science from Rovera's time in the 1950s has dropped dramatically to a critical level. And unless you guys can be persuaded to help invest in that area, I think we're in deep, deep trouble. The other reason why that's so important is because of these new technologies. We can look at any soil, and we do this from soil passed from New Zealand, America, flown into our labs. We can put them in front of the CAT scan, and within approximately 3 minutes, we can tell everything about the porosity of the soil, how connected it is, how much is there, how tortuous it is, and from that, we can start predicting almost any process within the soil system. This technology has only come to pass in the lab within the last 5 years, and there's already, I think, 3 in Australia, and it's a fantastic bit of technology which we can take real soil from real soil systems and tell you almost everything about it in terms of the physics, and it's a very exciting process. The other thing that I know you guys have been investing heavily in is soil biology, and I spent, I think it was, five years ahead of one of the largest groups in soil biology, mainly microbiology, in the UK. And it's taken many, many years to get the technology platforms up and running in terms of the genetics, and they've done a tremendous job there, so much so that prior to that, only 1% of the total population of the microbiology was able to be seen in the lab, because we just could not come up with culture methods. Now with the molecular technologies, we are able to see almost 100% of them. And where they've worked tremendously well is in soil pathogens. And where they're trying to put some effort into is connecting those numbers with function. But there is a challenge. The challenge is this: in a handful of fertile soil, there's more individual organisms than the total number of human beings that have ever lived. Pick up a handful of fertile soil, and you have that amount of organisms in your hand. So what? Because so what's the huge question. We can count and count until the cows come home. We can produce papers that please our university masters more than you would imagine, but it has no relationship to function. Absolutely none. If we take a sugar cube of soil, and this is a root from a plum tree grown by Albert Trauss in 1943. Sugar cube of soil, make assumptions about the clay contents. Roughly you've got about 20 metres squared of surface area in there. And then ask the question, how many, in that gram of soil, in that sugar cube of soil, how many protozoa, bacteria and fungi do we have? Well, in a very fertile soil, 10,000 protozoa, 10 to the 7 bacteria, that can go up to 10 to the 10, 5 kilometres of fungi. I want to ask you a question, and if you can come within an order of magnitude of it, then you'll win a drink from a Scotsman, which is a rare occurrence. What percentage of that surface area there is covered by these guys here? So, who says 100%? Hands up. 50%. 10%. A few more hands. 100%? One hand up, he's wrong. 5%. 1%. 0.1%? I think people have been reading my papers. Roughly, you've got 10 to the minus 6% of the total space covered. You might think that's tiny. Actually, it's quite large. If you think of the population on Earth, what percentage of the surface area do we cover? It's actually 10 to the minus 5%. Microbes, I call them bugs, act in a very similar way to you and I. They like food, they like sex, they like resources, and they run away from predators. And there's very few areas where all of those can come together and they congregate around those areas. What this measurement tells you is that in terms of soil biology, the key factor is the physics of the soil, not the soil biology. The physics of the soil forces all the biology to congregate in certain areas. And the physics controls the water flows and controls the movement, because most of these guys, all of them, rely on water. And what we have just now is an amazing array of biology in soils. And this is techniques that myself and a few colleagues developed in the UK. Over here we have pesticide-degrading bacteria funded by Bayer many years ago. The question was, if the bacteria are there compared to there, what's the probability they'll degrade a pesticide? So we did a lot of mathematical models for them. This is a nematode, Melodogine incognatia, which is a pathogen. Here is a naked amoeba, which is a shape changer, accessing small-scale pores to eat bacteria. And this is fungal spores sitting in a root, and you can see the direction they have, all pointing to the outside. The interesting thing is they know where they are and they know where they want to go. And so what the point I'm trying to make here is counting them is virtually useless. Telling me what species are there is not that much better. It's just not that much better. I want to know where they are and what they're doing. That's the biggest question I've been asking for the past decade. Where are they? What are they doing? How can I stop them? How can I help them? And that's the question the farmer will ask. Now we have a huge amount of biodiversity and life in soil. With the technologies, we can count them, just. The key thing is we have to stop training kids to actually use these machines. These machines, the molecular genetics machines, are actually very easy to use. There's recipes where you use them. We have to train kids to start thinking of the importance of that biology to the function in soil. And I know you've got the GRDC Beneficial Soil Microbes program. A big suggestion from me there is to accept that we can now count them, move on from that, and put effort into training younger scientists how to think in terms of what the farmer needs, and how to think in terms of the fundamentals of the soil. Because there's a lot of emerging science coming that will not easily directly go into E, but that emerging science over 20, 40 years, a small percentage of it will pop up and change our lives radically. And they will change the business of GRDC radically. And if GRDC takes his eye off that, and doesn't put a small portion of money into that emerging science, that crazy science, adventure science, then I think we're in trouble, and I think that's badly needed just now. So we have to stop doing what we call the biology of numbers and differences. We have to relate soil biology to the function, and I've got two examples of how we do that. And the first one here is a cube of soil, and it's got triticale growing in it, and in the middle it's got phosphorus granule. The key question we were going to ask is, with our new X-ray technologies, can we literally see the roots and count them? The answer appears here, without getting into detail. With using a normal way of counting, literally counting 1, 2, 3, comparing it to a non-destructive way that takes 4 minutes to scan rather than days, we have a fantastic relationship. And I want to show you, and I think you'll be one of the first group ever to see this, of how you count roots. Real soil, real roots, a band of pea fertiliser put in there, there it's coloured. Nothing is false except the colour. And we can see the roots, we can see the response of the roots. So now we have access to the hidden half of the plant that the plant breeders have ignored for years. They've ignored for a very good reason. If you read the paper I've written, you'll understand why. I would have ignored them. But now we have the technology that reveals that hidden half. We have the technology that we can put any plant in any soil. This is a vertisol. Any plant, any soil, any treatment, and we can, using this technology, tell you the response of the plant roots. We can tell you the important traits to look for in terms of the breeding, and that to me becomes incredibly exciting. And here's some of the first answers. This is unpublished work we're doing with Chris Guppy in the room and Richard Flavo, and you'll be pleased to hear that GRDC support Richard in this work with a scholarship, and that's been a small amount of money, and the returns I believe have been huge. And basically we can see what we should have seen, and we have a massive response of the roots here. The difference is, instead of weeks and months to try and get this, that scan took about, I think it was 12 minutes to do. A tremendous opportunity for GRDC and a tremendous opportunity for the scientists. The cool thing is that this hidden half has been lost to us. My first job in SCRI, Scottish Crop Research Institute, was to dig out potato roots and count them. That's really why I did 15 years of university, to dig roots and count them. But now we don't need to do that. The plant readers can focus on the largest part of the plant, but also we can look at the tops. This took a minute to scan. If anyone's interested what goes on inside, you can see and you can count. This is what we're doing with Cyril Plant Industries. And I, again, think it's very important. So one technology, a couple of changes in mindsets, bringing soil, plant, roots, and top together. Really, we've opened up the Pandora's box now. The opportunities are so big that we find it hard to focus because they are so large. And a great focus can actually be done through GRDC. The other big question that we've got is that how do roots and microbes help water uptake by plants? This is not a question that many people have asked. Apart from a bushkill mycorrhiza, and that area is still unproven actually. Because one of the important things is this little curve. I call it the most important curve in all of ecology in the world. This is the, as you all know, moisture retention curve. Here we have the energy required to take water out, and here we have the water in the soil. And as you increase the energy of the suck, you can see the water depletes from the soil. But the plants, and this is from wheat plants, have a very neat natural way of actually grabbing more water out, but not using any more energy. And that's the crucial thing. No more energy, better water uptake. And what they do is they release a surfactant, much like fairy liquid, which reduces surface tension and contact angle in the soil. And that means for the same energy expended, you pull out far, far more water. And this happens to be called lecithin here, but you can see the marked difference between the water left, where there's no plant root, and the water left, where there's plant roots and surfactants. The other added unknown bonus to this was that it had a massive impact on pea absorption. Here's the solution pea concentration here, and pea absorption here. You can see that, without getting into too much detail, every time there was a surfactant, the plant was able to take out more phosphorus. A naturally occurring trait within a plant that now the plant breeders can look at. We can actually see both things ongoing in the tomography because we can see water as well. And suddenly, if we can capture the controlling factors of the release of this surfactant from natural compounds, we can then upregulate, downregulate, and manage to breed more plants that look at where the action is, and most of the action is in the soil from the roots. And the other opportunity is bacteria in the soil. These bacteria are called Pseudomonas fluorescens. There's many in the soil. And here we have the surface tension. So all you need to know is, as that goes down, for the same energy, you get far, far more water out, far, far more phosphorus out, okay? Here are different types of Pseudomonas fluorescens. And you can see that the different types have a massive change in surface tension. To give you some context, water, pure water, is up at 72, okay? And when you look at lecithin, this is what the plant root can do, and it turns out that the bacteria can do far more. Can you imagine a product now, not just a plant breeding product with a new line coming in, but a product that you can add to the soil system that's inherent in the soil system anyway, so competition becomes less of a problem? That actually, by adding to the system, helping the soil biology, increases water uptake and pee uptake. I think it's incredibly exciting. And this is what happens in this moisture release curve. Here's the suction or the pressure. Here's the volumetric moisture content. Up here, it's a saturated system. Air enters, large pores empty, then the small pores start to empty. The other neat thing that this does is that this is a valve that controls all air entry. Obviously in a saturated system, no air's going through. But when the large pores decrease, connected air spaces happen. Why is that important? Because that's how greenhouse gases get out of the soil system. This moisture release curve controls everything in the soil, and there's less than 1% of soil scientists use it, because what they do is they concentrate on this moisture content. It's got nothing to do with the moisture content. It's all about the energy in the soil. Everything's about the energy in soil. And that's why if you really want to know about soil, you stick tensiometers in, not capacitance probes, right? The capacitance probes gives you some indication of that. The tensiometer tells you everything about the energy. And you can take that and use it across a wide range of soils. It's a diagnostic. And as advisors, a key thing for all soils, all plant growing material, is the construction of this curve. So with these aspects where we're looking at natural products from plants and roots and bugs, we can actually now contemplate designing large-scale farm soil systems. That is my vision, to design it to our purpose and to our requirements. The CRC Polymers, which many people have noted have been awarded a 4th round, most people hadn't realised it's probably the largest sum ever given to soil plants through the CRC, 14.6 million, with the largest theme in water and food security. And their main aim is to develop this new range of polymers to help farmers. The key aspect of it is water security. UNE is a key researcher on the physics, Plant Industries are a key researcher on the plants, and UWA are key researchers on the soil biology. And that ability to connect those great, I would call excellent teams, is exactly what GRDC should be doing. Going out there, finding who's the best, and connecting them up with some funding. It's the key thing. This starts in July. GRDC have funded this to the tune of, I think it's $300,000 a year for the next 5 years. So you should be expecting stuff to come out of that. But it becomes incredibly exciting when you do that. And my ending thing is a funding priority for Australia, and not only for GRDC, because you would not be able to do it at all. I think that soil science has been neglected for the past 50 odd years in Australia, just at the time when Europe has actually increased funding. It's not all about soil carbon. Jeff will chat about that in a minute. It's about productivity and sustainability. And I think we have to look at these neglected areas. It does lead to better management. It does lead to what we call ecosystem function. It does relate to cutting greenhouse gases. But the key thing, ladies and gentlemen, is it must relate to food productivity. Thank you.
Iain, what do you think about mining in these fertile valleys, and what influence can the two industries exist together without jeopardising the soil security as you talk?
Thanks for that. And I don't pretend to know much at all about the mining industry here. I wouldn't comment. All I would say is if you've got fertile soil, the first and foremost thing is the precautionary principle. It must be the first and foremost thing. We have to protect the fertile soil. Now, if there was some way to combine it through a lot of research, and I mean a lot of research, not one-off investigations, then perhaps we can combine it, but the precautionary principle applies. The Deans of Agriculture of Australia put out a call for a moratorium on certain aspects of mining, primarily because of the precautionary principle, and I think we should not deviate from that one iota.
Iain, you seem to be going down the route at the end of adding microbes or adding things to manage microbes. We seem to have failed miserably in that area in the past. Why is that going to succeed in the future?
So the reason we've failed miserably in the area is we've not had that connectivity between the chemistry and physics and biology. And we've had a lot of snake oil salesmen out there saying, all you need to do is add this and then everything will be okay. Prevalent initially in the sports turf area, where they've got large and intensive non-agricultural grasslands, but spreading now into agriculture. A key thing is the appropriate tests in the appropriate labs. I know it slows things down, but really what you're looking at is a sustainable and reproducible result. And I truly believe that what we're trying to do is mimic what happens in the soil now with a far greater understanding of the science behind it, the fundamental science, which simply has not happened before. I've done a lot of work in sports turf, primarily golf courses. I used to live about 18 miles from 100 courses in Scotland. I played most of them, I tell you. But we did a hell of a lot of work out there. And almost every occasion we went and visited, either someone was there offering the groundsman a bag of something, solve all his trouble, or he'd just been. And we were always confronted by this. And it became obvious, and that's one of the reasons I set up soilcolumn.com when I came here, is to actually put the evidence out there. Because I don't think there's any magic bullet without a full understanding. And I would include biochar, glomalin within that whole thing. So I think we understand it far better. Therefore, the risk is lower and the chances of success are higher.
Iain, you talked about a vascular mycorrhiza, and some potential doubts over the science of that. Could you expand a bit more on that?
Sorry, it was just in relation to water uptake there. It wasn't in relation to the science. It's quite there's quite varied responses in that. And the answer to the question, does a vascular mycorrhiza significantly help water uptake? So it wasn't in the science, but in the variability that we found. I've done a lot of work on a vascular mycorrhiza. Happy to talk about it later, primarily related to glomalin.
Iain, one of the key issues I think we're running into in agriculture is that this concept that everyone has a right to cheap food, and the cost of inputs isn't going down, but the cost of food is, the cost we're getting from producing food isn't going up. So, from a very practical perspective, it's very hard to put more inputs in terms of nutrients when you're not being paid for that in terms of the value of the product.
Well, you're not wrong, but as we deplete our phosphorus reserves in the world, as the price of inorganic fertiliser increases, we don't have much option to look at alternatives, and there's a cost to everything. And I think the aim of the CRC would be, I guess, because it's just starting, is to ensure that any products come out are affordable by farmers, because it's companies like Bayer, companies, I used to call them Rhone Poulenc, those agrochemical companies are the ones going to produce it, so they need to be cheap for you to use.